Welcome! We are beginning Chapter 24's section on metabolism. Metabolism is the sum of all the biochemical reactions inside a cell which involve nutrients. Metabolic processes are either anabolic or catabolic. Anabolism is the synthesis of larger molecules from smaller ones, like the synthesis of proteins from amino acids. Catabolism is the hydrolysis of complex structures to simpler ones, like the breakdown of proteins into amino acids. Cellular respiration is a series of catabolic reactions where food fuels are broken down and the energy from the food is captured to form ATP within cells. ATP is our cell's energy currency that is used to do cellular work. As ATP is phosphorylated, energy can be used. Phosphorylation is when enzymes shift high-energy phosphate groups of ATP to other molecules, releasing the cellular energy. Phosphorylated molecules become activated to perform cellular functions. There are 3 stages in the processing of nutrients. Stage 1 involves digestion, absorption, and the transport of the absorbed nutrient to the tissues. Stage 2 involves cellular processing that occurs in the cytoplasm. There are anabolic synthesis reactions which make lipids, proteins, and glycogen from the delivered nutrients. And there are catabolic reactions which break down the nutrients through glycolysis into pyruvic acid and acetyl-CoA. The 3rd stage occurs in the mitochondria. Intermediates are oxidatively broken down into carbon dioxide, water, and ATP. Cellular respiration includes the glycolysis of stage 2 and all of stage 3. Figure 24.3 illustrates the 3 stages of metabolism of energy-containing nutrients. The first stage occurs in the GI tract. Nutrients are digested into absorbable units, then absorbed into the blood, and finally transported to tissue cells. In this stage, proteins are broken down into amino acids, carbohydrates are broken into glucose and other sugars, and fats are broken down into glycerol and fatty acids, all by catabolic reactions. The second stage occurs in our tissue cells. Newly delivered nutrients are either built into lipids, proteins, and glycogen by anabolic pathways, or broken down by catabolic pathways to pyruvic acid and acetyl-CoA in the cell cytoplasm. Glycolysis is the major catabolic pathway for this step. The 3rd stage occurs in the mitochondria and is almost entirely catabolic. It requires oxygen and completes the breakdowns of foods, producing carbon dioxide and water and harvesting large amounts of ATP. The citric acid or Krebs cycle and oxidative phosphorylation are the major pathways for this stage. How do our cells capture energy liberated during cellular respiration to make ATP molecules? There are 2 mechanisms: substrate-level phosphorylation and oxidative phosphorylation. Substrate-level phosphorylation occurs when high-energy phosphate groups are transferred directly from phosphorylated substrates to ADP. Essentially, this process occurs because the high-energy bonds attaching the phosphate groups to the substrates are even more unstable than those created in ATP. ATP is synthesized by this route twice during glycolysis and once during each turn of the Krebs cycle. All the necessary partners for these reactions are found in the cytosol for glycolysis and the mitochondria for the Krebs cycle. This figure illustrates substrate-level phosphorylation, where an enzyme catalyzes the reaction of a phosphate group leaving a substrate for ADP. ATP is created along with a new product. Oxidative phosphorylation is a more complex process and produces the most ATP. Oxidative phosphorylation is carried out by electron transport proteins embedded in the inner mitochondrial membranes and is an example of a chemoosmotic process. Chemoosmotic processes couple the movement of substances across membranes to chemical reactions. In this case, some energy released from the oxidation of food is used to pump protons, written as hydrogen ions, or H+, across the inner mitochondrial membrane, creating a steep proton concentration gradient. As protons flow back through the ATP synthase membrane channel protein, energy from the flow is used to phosphorylate ADP. This figure illustrates oxidative phosphorylation. Protons are pumped across the inner mitochondrial membrane using the energy from food. This develops a steep proton concentration gradient. Then the protons want to flow back across the mitochondrial membrane. When they do this, they go through the ATP synthase channel protein right here. The ATP synthase captures the energy of the protons moving across the membrane and then uses it to add a phosphate group to ADP, creating ATP. All right, let's move on to carbohydrate metabolism. All carbohydrates eventually are transformed into glucose. Glucose enters tissue cells by facilitated diffusion, a process that greatly enhanced by insulin. Immediately upon entering the cell, glucose is phosphorylated to glucose 6-phosphate. Most cells lack the enzymes to reverse this reaction, so it effectively traps glucose within the cell. Because glucose 6-phosphate is a different molecule from simple glucose, the reaction also keeps the intracellular glucose levels low, maintaining a concentration gradient for glucose entry. Only intestinal epithelial cells, kidney tubule cells, and liver cells have the enzymes needed to reverse this phosphorylation reaction. All catabolic and anabolic pathways for carbohydrates begin with glucose 6-phosphate. Glucose is catabolized using the reaction displayed. Glucose plus oxygen produces water, carbon dioxide, 32 ATP, and heat. Glucose breakdown is complex and involves 3 pathways: glycolysis, Krebs cycle, also known as the citric acid cycle, and the electron transport chain and oxidative phosphorylation. This figure illustrates how ATP is formed in the cytosol and in the mitochondria of cellular respiration. Glycolysis occurs in the cytosol and breaks down each glucose into 2 molecules of pyruvic acid. The pyruvic acid then enters the mitochondrial matrix where the Krebs cycle oxidizes it into carbon dioxide. During glycolysis and Krebs cycle, substrate-level phosphorylation forms small amounts of ATP. Energy-rich electrons picked up by coenzymes are transferred to the electron transport chain, built into the inner mitochondrial membrane. The electron transport chain carries out oxidative phosphorylation, which generates most of the ATP in cellular respiration. Let's start with the first pathway, glycolysis, also known as the glycolytic pathway. It occurs in the cytosol of cells, involves 10 steps, converts glucose to 2 pyruvic acid molecules, and is an anaerobic process, meaning that it does not use oxygen, but it can occur in the presence or absence of oxygen. There are 3 major phases of glycolysis: Phase 1, sugar activation; Phase 2, sugar cleavage; and Phase 3, sugar oxidation and ATP formation. Beginning with Phase 1 of glycolysis, sugar activation. During this step, glucose is phosphorylated by ATP to fructose 6-bisphosphate. Fructose 6-bisphosphate is phosphorylated again by a second ATP, resulting in fructose 1,6-bisphosphate. This phase is considered the energy investment phase because an input of 2 ATP was required. These 2 ATP were used, but they will be recovered later. This phase provides the activation energy needed to begin the latter pathways. This figure illustrates glycolysis' first step, sugar activation. Here, 2 ATPs are invested to phosphorylate glucose into fructose 1,6-bisphosphate. The second phase of glycolysis is sugar cleavage. Fructose 1,6-bisphosphate is split into two 3-carbon fragments. The fragments can interconvert into one of two isomers, dihydroxyacetone phosphate or glyceraldehyde 3-phosphate. Here is phase 2 of glycolysis of sugar cleavage. Fructose 1,6-bisphosphate is split into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Phase 3 of glycolysis is sugar oxidation and ATP formation. Phase 3 consists of 6 steps with 2 major events. First, each 3-carbon fragment is oxidized by removing a pair of hydrogens, which are then picked up by NAD+, forming the reduced NADH plus H+. The hydrogen carries a portion of the glucose's energy. Second, inorganic phosphate groups are attached to each oxidized fragment by high-energy bonds. Later, these phosphates are split off. Enough energy is captured to form 4 ATP molecules in a process called substrate-level phosphorylation. This figure shows the third phase of glycolysis called sugar oxidation. In this phase, the 3-carbon fragments are oxidized by removing hydrogen, and 4 ATP molecules are formed, as well as 2 pyruvic acid molecules. Here we see the entire glycolysis process. Please make sure to study this figure. Although 4 ATPs are made, 2 ATPs were invested in phase 1, so they must be subtracted. So the final products of glycolysis are 2 pyruvic acids, 2 reduced NAD pluses in the form of NADH plus H+, and a net gain of 2 ATPs. For glycolysis to continue, more NAD+ must be present to continue to accept hydrogen atoms. The supply of NAD+ is limited. NADH must donate its accepted hydrogen atoms to become NAD+ again to be free to pick up more H+ from glycolysis to continue. If oxygen is present, the NADH will transfer its hydrogen to proteins in the electron transport chain within the mitochondria. If no energy is present, NADH will give hydrogen atoms back to pyruvic acid, resulting in lactic acid. Some lactic acid leaves the cell and is picked up by the liver, which can convert it back to glucose 6-phosphate. Some lactic acid may be oxidized back to pyruvic acid when oxygen becomes available, and then enter the aerobic pathways. Prolonged anaerobic metabolism can lead to acid-base problems. Glycolysis results in faster ATP production than aerobic respiration, but it yields far less ATP. This figure shows all 3 phases of glycolysis. As a review, phase 1 is sugar activation when 2 ATPs are invested and glucose is converted to fructose 1,6-bisphosphate. Phase 2 is sugar cleavage, where the fructose 1,6-bisphosphate is broken into 2 3-carbon pieces. Phase 3 is sugar oxidation and ATP formation, where the 3-carbon fragments are oxidized and 4 ATP, 2 pyruvic acids, 2 NADH plus 2 H pluses are formed. Glycolysis can occur with or without oxygen presence. Okay, we're going to stop here and pick up this in our next segment.